All right, I think we've given enough time now, so we're going to go ahead and get started. Good morning, good afternoon to everyone. This is Tim Wesselman. I am the CEO and founder of Rosalind. It is my pleasure today to kick this off and hand you off to the very capable hands of Dr. Ryan Friese, who's our Vice President of Customer Experience. Before we get started with the session, I have a few brief comments to set the stage for today. First, I just want to comment that I am extremely excited that we're doing this series. Ryan has been collecting 10 years of valuable knowledge and best practices from his experience with NanoString and working directly with customers like you, scientists like you, helping you work with the instrument, helping you work with your assays, and helping you get answers out of the data in ways you need to. And Ryan is an absolute expert in this space. These are the experiences and the knowledge that he's developed over these years that he's always wanted to share. Normally, we reserve classes like this for our professional and our enterprise customers. We've opened this up to everyone. And so we wanted to make sure we brought this incredible value to you. Now, I'm going to cover a few quick housekeeping points, and then we'll come back to the session. So first, as a reminder, this is a webinar. You are all muted. There is a place to capture your questions. Please do. You can click on the question box on the right, type in the questions you have. We get them as we're going. I'm also here on the line supporting the session with Kuki, who is our Director of Product and Business Strategy. She and I will be gathering those up and preparing responses either during the session or as we get to the end, and we'll bring those to Ryan for you. So please don't wait till the last minute. This has been very typical with the sessions in the past. I know you all are really enjoying these. The feedback has been outstanding, and there are hundreds of you here today. So please don't wait on asking questions. There are some really useful handouts also. So click on that box on the right. Feel free to download those. Some great resources where you can learn more. With that, I want to come back to some details about this. So Rosalind is a strategic partner of NanoString's, now Bruker. We really appreciate the strategic partnership. I personally love the Encounter instrument and also what NanoString has done in spatial with geomics and cosmics. But I think there's a couple things that we lose sight of. And I'm spending a lot of time with scientists like you, as is my team right now. As many of you know, there's some transitions going on with our starter subscriptions, bringing you all to our new professional capabilities. But in all that conversation, I think what I hear a lot is there are folks that over the years have forgotten how powerful it is to be able to have a targeted panel and the ease of use that the Encounter instrument provides. And I just wanted to take a moment and just emphasize, I think that this is one of the moments for scientists to be able to have a 15-minute hands-on prep and be able to get results that Rosalind takes you to the answers to find in minutes. And having that end-to-end directly in your control experience is why I refer to these as the scientist instrument with the scientist platform. So just really want to emphasize the importance here. Now, we are going to go into pathway analysis today. Ryan's going to go deep and discuss all of this. And while we go through that, this is a core strength of Rosalind. You're going to hear more about the 50 plus knowledge bases, our intelligence, investments, and capabilities of the platform, and how this goes far beyond anything that was ever available with NSolver, and which NanoString had provided. I think you all need to definitely keep in mind some of the importance of collaboration, enhanced security, and an ever updated experience for which Rosalind provides in comparison to the now decade-old open source solution that NanoString had provided in the past. So Rosalind is the preferred and premier solution for NanoString analysis. Our new geomics offering is coming right around the corner, and we look forward to introducing a masterclass on that in the days to come. And on that note, I've said everything that I should be saying at the beginning, so I'm going to hand it off to you, Ryan. Take them away. We're all looking forward to your session on pathway analysis. Thank you, Ryan.
Thank you, Tim. I appreciate that introduction. So I'm going to jump right into it and looking at a list of the attendees, and nice to see some familiar names. Thank you for joining these additional sessions, and welcome to everyone that's new for today's session. Yeah, as Tim mentioned, I've invested a lot of my career in Encounter NanoString knowledge in the technology and the data analysis course, which is why we're here. I originally started my career many years ago because I wanted to help people. I wanted to use my expertise, my knowledge, my learning to basically help human health and well-being throughout the world, which perfectly aligns with Rosalind's mission in general. So at Rosalind, we're creating this platform to help all of you discover and find out new things about all the different areas of research, science, and medicine that you're investigating. So we developed this masterclass to push forward all of the scientific research on the nCounter side. And so we developed this masterclass for 4 primary reasons. So we want to make sure that you can understand the value, but also the synergy when you pair Rosalind and nCounter together, all of the great things that can happen when they're paired together. I want you to have a deep understanding of the analysis principles and algorithms. So today we're talking about pathway analysis. In my experience, there's a lot of confusion about what those GSA scores means for nCounter. How do I interpret them? What do they mean? So I want to show you the math behind them. It's actually very simple arithmetic. So if they seem confusing in the past, I want to demystify that for you today so you can fully and confidently interpret the results. And then we'll get some new insights. So by pairing your targeted nCounter data with Rosalind, you can actually find novel insights by leveraging Rosalind's intelligence capabilities. So the whole session, or I should say the whole schedule for the masterclass is shown here. Today we're on pathway analysis. If you didn't get to attend previous sessions, we do have recordings available, so take advantage of those. And then we'll be finishing by the end of June with cell profiling and meta-analysis. So I won't belabor the points of nCounter technology and why it's so valuable for the scientists, but we all appreciate its speed to results. Get your data within 24 hours, very little pipetting in the lab, so it's very minimal bench work. My personal favorite part about the technology, it's very elegant and simple in terms of the instrumentation and chemistry, and all of that lays the foundation for the digital precision. So we know it's enzyme-free, single molecule counting, very little sources of technical variation. It's highly reproducible, and it's best in class with the graded RNA samples. But of course, today we're talking about data. So from the data perspective, I love working with nCounter data because the raw data files are small. RCC files are just on the order of kilobytes. Very easy to transfer and store the data. It's very rapid to process the data. You don't need a specialized high compute processor to process the data. And it's easy to interpret because it is intuitive, it's single molecule counting. So again, I'll just emphasize this last bullet point here. At the end of today's session, I want you to walk away with an understanding of the NanoString pathway analysis and how it works. Because if you don't understand how the math works, then you could potentially be limited in how you interpret the results. So that's the focus of today's session. We're going to be delving deeper into the Rosalind platform more than we did in previous sessions. Last session, we looked at the quality control module. Today, we'll be looking at the differential expression and pathway analysis modules in more depth. And then in the future, we'll also be looking at cell profiling. If you weren't aware, we recently launched some additional capabilities for nCounter specifically. So some of these were the focus of our previous masterclass sessions: custom pruning, custom normalization, pathway analysis, of course, we're talking about today. And we'll start getting into some of the knowledge and intelligence capabilities of the platform as well. But in my experience over the past 10 years, Rosalind is the place to analyze your nCounter data. It's the most powerful and collaborative solution on the market. So before we get into pathway analysis, I'll summarize with one more figure here. And this is showing you the synergy between nCounter and Rosalind. So it's a complete end-to-end workflow. You start in the lab with the chemistry, setting up your hybridization reaction. It takes you 15 minutes of pipetting to set up the reaction. Load it in a thermocycler overnight, come back the next day, load it into your instrumentation. You go from sample to your raw data in less than 24 hours. And at that point, you load it directly into the Rosalind platform. And you can take advantage of all the knowledge and intelligence capabilities to enhance the value of your targeted panel, to extend the utility of your targeted data. So that is my introduction. And now let's start talking about pathway analysis. So like I've done in previous sessions, I want to pose a question to you to start stimulating your thinking. Let's get back to basics, back to first principles. I'm going to pause for a few seconds here, but think about why we are performing pathway analysis. Just take a few seconds here and think about why you want to do this in your own analysis. Okay, so what I came up with was actually a nice quote from a publication that I found that I was reading about the other day. So pathway analysis, at least at the highest level, in my perspective, is represented here with this quote. So it's a set of widely used tools for research in life sciences intended to give meaning to high throughput biological data. The methodology of these tools settles in the gathering and usage of knowledge that comprise biomolecular functioning coupled with statistical testing and other algorithms. This is a very high level definition, but I think it highlights two nice things. The sentence at the top is that you need to get data, you need to extract data out of your high throughput multiplex data. nCounter is 800 genes, it's not whole transcriptome, but if you've ever stared at a spreadsheet of 800 genes, 800 rows, it's really difficult to get meaning out of that spreadsheet of data. So we're applying pathway analysis to extract as much information as possible out of your nCounter data, out of your targeted panel. And the second sentence down there is that we are gathering knowledge. We're gathering and using knowledge about the genes and their memberships in different pathways. So we're trying to assign functional roles to all of our different genes in our data set and inferring some additional second order pathway level information or even drug level information or cell types, that kind of thing. So it's really get as much as we can out of our data by learning about our individual genes and getting these higher level inferences. Rosalind is a perfect place to do this because to your targeted data, we're bringing a foundation of knowledge and some intelligence capabilities that you may be missing out on if you're not using Rosalind. So let's take a look at how we approached the pathway analysis in the Rosalind platform. When we're thinking about developing this specifically for nCounter, what did we do? So we looked at what was the current state of affairs. So many of you might have used NSolver in the past, which is NanoString software. So we know in NSolver Basic, pathway analysis is not available. NSolver Advanced Analysis, we know that in that software package, you can apply about 24 or so pathways to each of the different targeted panels you're analyzing. These pathways were curated by the wonderful NanoString bioinformatics team. They leveraged their expertise, utilizing public data, et cetera, and their own specialties to curate essentially 24 or so pathways for each of these off-the-shelf panels. So that's what's available in advanced analysis. Now at Rosalind, we wanted to make your targeted data even more valuable and extract as much information and get novel insights out of your data. So we added a lot of new features. Essentially, we went to public repositories, public knowledge bases, and we pulled out thousands of new pathways, terms, and signatures associated with diseases, drugs, cell types, virology, a lot more information. So in addition to those 24 or so pathways you're analyzing in NSolver Advanced Analysis, we're bringing thousands of more terms to your pathway analysis to give you this very expanded, extensive approach to maximize that data, maximize that level of information you can extract from your targeted panel. And to do that, we leverage Rosalind Intelligence, and I'll show you that in a few minutes. So all that's summarized here on this slide. And there's two things I'll point out here, which are kind of exciting for me. I'm a big data analysis nerd. But previously in advanced analysis, there were no p-values associated with any of the pathways because you weren't performing enrichment or any type of statistics. So now in Rosalind, you can get these p-values, which have been a long request by NSolver customers or nCounter customers. So you can now get p-values for your pathway analysis. So let's take a look at what some of the pathway analysis might look like. If you've performed this type of analysis on nCounter data before, then you're familiar with NanoString's GSA scores or gene set analysis scores. So on the left-hand side is what this might look like in the Rosalind platform. But what we're showing you here is just a selection. These might be one of those or a handful of those dozen or so or 24 pathways that the NanoString team, bioinformatics team, expertly curated for each individual panel. So I believe this was from immunology-based panel. So for each one of these curated pathways, you get this significant score. So this is a score associated with a pathway, but the question is, what does this mean? I found there is much confusion around this. It's not very intuitive on how to interpret these. So I want to break it down for you so we can fully understand. On the right-hand side, these are excerpts from the manual, the advanced analysis manual from NSolver that describes these pathways, how they're calculated and what they mean. I know that not everyone reads the manuals, so I wanted to just pull this information and put it on this slide, and let's talk about this and then break it down so we can have a full understanding of what these scores mean. Because at face value, these really don't have any meaning by themselves. There's no p-value associated with them. It's really hard to interpret. So if you're brand new, let's take it from the basics and slowly build up from the foundation. First thing to notice is for these GSA scores, which were developed by NanoString again, there's two types. There's a global score, also called undirected, and there's also a directed score. So I'm just going to read this to you. It's from the manual if you haven't read it before. But for the global score calculation, it's done like this. Differential expression analysis calculates a t-statistics for each gene against a covariate in the model. A gene set's global significance score for a covariate measures the cumulative evidence for differential expression of genes in a pathway and is calculated as the square root of the mean squared t-statistics of genes. So in case that's not clear for everyone on the call, I want to break down the math because this may sound complicated, but it's simple arithmetic. So I'll show you what this looks like. The directed score below, again, I'm just going to read this to you in case you haven't read this before. The directed global significance statistic is similar to the global significance statistic, but rather than measuring the tendency of a pathway to have differentially expressed genes, it measures the tendency to have over or under expressed genes. It is calculated similarly to the undirected global significance score, but it takes the sign of the t-statistics into account. So again, this is describing kind of how this score is interpreted, how it's calculated, but it's not always clear to all the readers that see this for the first time. So let's break it down and go a little deeper on what this means. So as I've done in the past, I'm going to get out of PowerPoint here, and I'm going to break it down for all of us in Excel. So give me 1 second as I transition over. Okay, so let's take a look at the global score, the global GSA score on the left, and the directed score on the right. Some of us might be very intimidated or apprehensive by these equations, which might look like a foreign language. So, but again, these are basic arithmetic. So let's break them down slowly one by one so we can fully understand the interpretation of these scores. So we'll start with this global score on the left. So what's involved in this score is it's something called the t-statistic. P, which is the total number of genes in a pathway. And one thing I want to emphasize before we get into the math is when you're calculating these scores, either the global score or the directed score, we're looking at every single gene of the pathway. It's independent and regardless from the p-value. So in another type of approach, looking at pathway analysis, such as pathway enrichment, which I'll show you a little later, with this pathway enrichment approach, you are focused only on the genes with that significant p-value, let's say p less than 0.05. Those are only the genes you're concerned with. But in both of these scores, global score and directed GSA score, you look at every single gene in the pathway, regardless of the p-value. So that's the first thing to remember here. Many people are not aware of this, but it looks at every single gene in the pathway, regardless of statistical significance. So hopefully that's clear and that makes sense. So let's imagine a hypothetical scenario where we are looking at, let's say, the adhesion pathway. Let's pretend there's these 6 genes in the adhesion pathway, and we want to perform a differential expression analysis between 2 groups of samples. Let's say we have some treatment samples and control samples. We want to find out which genes are significantly differentially expressed between treatment and control. So the output of that, as many of us know, is a p-value indicating is the gene significant. So that p-value for our genes is shown here. So some of these genes are significant, less than 0.05. These 2 are not, and then these 2 genes are significant. But remember, when we're calculating these scores, we are not looking at the p-values, we are looking at every single gene in the pathway. So what we do instead is we look at the t-statistic. Now, I don't want to get too down the weeds of statistical testing, but when we perform differential expression testing in Rosalind, we apply statistical tests, and the output of this is a test statistic. In this case, we're calling T, it's our test statistic. And then that test statistic is what's translated into the p-value. So you can think of this test statistic as being one step upstream from the p-value. So Again, we're not looking at the p-value, we're looking at these test statistics. Now, an important thing here is that this test statistic is going to be used to calculate our score. But if you don't know what this means, it's really hard to interpret the score. So let me provide a little guidance here. This is not necessarily true from a statistical or mathematical point, but conceptually, the way I think about this test statistic, it's basically kind of a summation of the fold change of a gene. So it's not, again, this is not a direct translation, but conceptually, I like to think about this number as how much fold change is there in a particular gene. So, these three genes here, ITGAL through ICAM1, they have this positive test statistic. So I think about them being upregulated in my treatment group versus control. These three genes at the bottom here, they have these negative test statistics. So I can think of them as have a negative fold change. In other words, they are underexpressed in treatment versus control. So just conceptually, and again, this isn't mathematically accurate. So if there's any statisticians listening, please forgive me. But just to provide you a framework on how to interpret these scores, think about this as looking at kind of the fold changes of your genes. So with that being said, let's look at how this global score is calculated. So the T statistic, this is TI. So all we do to calculate this formula is shown down here in this table. So the first thing we do is calculate the test statistic. Again, this is from the differential expression testing when we run our analysis. So that's generated 1 per gene. Then we take this, we square it. So it's your T squared. So that's shown here. So 79.57 is 8.92 squared. So and then 2.42 squared is 5.86. 1.06 squared is 1.12. So all these values here is just T squared essentially. So that's shown here. So that's represented here. And then this sigma, this is a Greek symbol sigma if you're not familiar. This is indicating summation. So it's telling us we just want to sum up all of these squared test statistics, all of these T squares. So essentially what we do, we just take all of these T squared and we sum them. So I just added all of these values together, all 6 values together, and that's 112.65. So that's all of this is saying here. We're just summing, this sigma is summing all of these T squareds. So that's 112.65. And then P, this is the total number of genes in our pathway, which happens to be 6. So the way that I think about this whole thing here in the parentheses, it's just the average of our squared T statistics. So essentially I took this 112.65, which is this here, and I divided by P, which is 6 genes in our pathway, and that's 18.77. So we're almost done. We've calculated 18.77. That is what's included in this parentheses here, and then we just take the square root, which is represented by this 1/2, this exponent of 1/2, and that's our score, 4.33. So that is how the score is calculated. It's very simple in terms of the math, but this nomenclature, this representation can be quite daunting if it's not easily understood and interpreted. So let's look at this global score here, 4.33. This is kind of the average value of all of our squared T statistics. The key here is that this is global, so we're not taking into account the direction. We're not looking at, is the gene upregulated? Is the gene downregulated? We're not looking at, are these T's, are they positive or upregulated? Are they negative or downregulated? We're squaring these values. So if you remember, we square all these T's, so everything becomes positive. So our global score is essentially this number that represents in the adhesion pathway, how much change is there in the pathway? Independent if the genes are upregulated or downregulated. Let's just look across all genes in the pathway, how much total change is there? And that, in this case, is 4.33. So hopefully that's making sense. If you're new to this, it might be a little daunting, but I want to continue on with the score on the right-hand side. So this is our directed score. And got to move a couple windows here. So it's calculated basically the same way. It's basically the same thing, but now we take into account, are these T statistics, are they positive, indicating upregulated, or are they negative, indicating downregulated? So that's where the directed term comes from. It's directed, indicating, are most of the genes upregulated in the pathway, or are most of the genes downregulated? So if the GSA score is positive, it indicates most of the genes in the pathway are upregulated. If the GA score is negative, it indicates most of the genes in the pathway are downregulated. So let me break this down in a little more depth. Now this gets a little crazy over here to interpret, but just bear with me. It's the same thing as what we just looked at, but now what we're incorporating is just the positive and negative signs. For positive for upregulated, negative for downregulated. So the interpretation should be very familiar to what we just looked at. So let's break this down in the math. I'm going to try to go through this slowly. Hopefully this is making sense for all of you. But we have our same 6 genes over here in our adhesion pathway, these 6 genes. One thing I want to calculate is what is the sign? So if the t statistic is positive, then I want to give a sign of 1, positive 1. If the t statistic is negative, then I want to give a sign of negative 1. So here we start to incorporate what is the direction of the change? Is it up or down regulated? We calculate the t-squareds just like we did on the left-hand side. So we take all of the t's, we calculate that squared value. And so that these numbers are the same as these. So it's the same t-squared, but now let's start to take into account this direction. So I simply multiply positive 1 or negative 1 to these t-squares, and that's the values here. So the positive ones, positive 1 times 79.57 is positive 79.57. Negative 1 times 4.71 is negative 4.71. So now we've incorporated the direction of the genes. Positive again here is meaning upregulated, negative is meaning downregulated. And now I do the same thing as I did on the left-hand side. I simply just add all of these values together, and those sum together is 60.44. And then just like I did on the left-hand side, I just want to take the average of this value. So it's 60.44 divided by the total number of genes in the pathway, which happens to be 6, and that's 10.77. This. So what I just showed you is what's represented in the parentheses here. I know it gets a little confusing if you're not used to this type of nomenclature, but that's exactly what this is trying to represent for you. So it's just everything in this parentheses here is equal to 10.07, which in this formula they call U for as their default here. So they're calling this value U. And so now we have U here. If we want to finish off this equation here, we have the value of U, it's 10.07. So there's 2 things we need to do. We need to figure out sign U, in other words, is 10.7 positive or is it negative? So that's easy to figure out. And then here, what we're basically taking is the absolute value of U and then taking the square root. So let's do those 2 things. So we take 10.07, which is U. Let's take the absolute value and then take the square root of it. That's 3.17. We figure out what is the sign of U. Is it positive or negative? In this case, 3.17 is positive. So we're assigning a value of 1 here. And to get our directed score, it's 3.17 times 1. So that, again, is one number that represents across all of the genes in the pathway, what is kind of their level of significance. And again, a good proxy for this is potentially thinking of this as type of like a fold change type measurement, even though mathematically it's not the same, but it's basically the direction. So the takeaway from me showing you all this is that we have the adhesion pathway, six genes in it, and we have this global score, which is overall all genes. If we don't look at the direction, if the genes are positive or negative or upregulated or downregulated, this is the number representing how much change there is in the pathway. When we start to look at the individual genes in this pathway and take into the direction if they're upregulated or downregulated, that's what this directed score is, which is 3.17. So that's the math. Let me break down some additional scenarios here to kind of illustrate how this is going to be interpreted. We're going to jump into the Rosalind platform because there's a lot of neat things I can show you there as well. But let me walk through a few scenarios here to illustrate how we can interpret these things. So I created a few different scenarios down here at the bottom. So the one we just looked at is there was a mix of genes. Some of them had positive T's indicating they were upregulated, some had negative. So it's this mixed bag of some genes that are upregulated and some that are downregulated. This is probably the most common scenario you're going to come across when you're looking at your real data sets. But there's another example here. Let's pretend all genes in this pathway are upregulated. In other words, all of these T's are positive. So let's use these and figure out how do the equations change here. So let's paste this in here, change the T statistics. And now what you'll see, I don't know if you realize this, but now the global score, 4.33, is exactly the same as the directed score. Now, if you think about what I just showed you, the only difference between the global score and the directed score is that we take into account the direction of the change. If the genes are upregulated, we give it a sign of +1. If the gene is downregulated, we give it a sign of -1. In this scenario, where all of the genes are upregulated in the pathway, the global score is exactly the same as the directed score, just because they're all positive. So let me show you another scenario, and let me ask you a question first. In this case, we have a scenario where every gene in the pathway is downregulated. I'm going to copy and paste these into this value here, and we're going to update the global scores and directed score. But what do you predict is going to happen? Think about this for a few seconds. In a pathway where all of the genes are downregulated, how is the global score going to compare to the directed score? Just think about that for a few seconds. So let me paste this in and show you. In this case, they're the same, but the signs are switched. So you remember the global score, this is independent of if the genes are going to be upregulated or downregulated. It just looks at all of the genes and say how much total evidence or how much change is there in the pathway. So we're not taking into account, is this gene downregulated or upregulated? The value of the global score is always going to be positive, and it's because mathematically we're taking the squared of the t. We're just squaring t. But on the right-hand side, we are taking into account, again, the direction. So all of these genes that have this, that are downregulated, get this assignment of -1, and that's used in the calculation here as well. So this is a negative score indicating that all genes in the pathway are downregulated, or that the gene is downregulated, I should say. Okay, let's go through 2 additional scenarios and we'll jump into the Rosalind platform. Here's a scenario, which is quite interesting. So what I did here, I had half of the genes upregulated, half of the genes downregulated, but they're perfectly balanced in that the upregulated genes are basically the opposite of the downregulated genes. So we have 5.32, 2.12, and 1.07. On the flip side, we have -5.32, -2.1, -2.12, and -1.07. So based off of everything I've just said, just take a few seconds again. And when I update the formulas, when I copy this information in, what do you think the global score is going to look like? What do you think the directed score is going to look like? And how are they going to look like relative to each other? Okay, let's take a look. Okay, so global score, as expected, this is positive. It's always going to be positive. We get a value for the global score. It's basically the average of all of these squared t statistics. It's 3.36. But on the directed side, did you guess that this value was going to be 0? Maybe you did, maybe you didn't, but the value is 0. And I presented this use case to you to indicate that kind of the minimum here is 0. A directed score of exactly 0 in this scenario indicates that all of the upregulated genes perfectly offset all of the downregulated genes. So that our directed score is 0. We have equal upregulation and equal downregulation. So that's why you get 0 here. And then let's look at one last scenario before we jump into Rosalind. This is what I called some very small non-significant changes. So let's imagine, so all of these t statistics, these are representing t statistics. These are really, really small. There's very, very little fold change here happening. So if I take this and I paste this in here, the same thing happens. So we have a global score. In this case, it's really, really small, 0.51. Our directed score is also 0 in this case. But I just want to contrast this in to our previous scenario here. In this scenario, we have all of these genes. Some are upregulated, some are downregulated. They offset each other. But if you look at the actual magnitudes of these changes, it's just really, really, really small. The global score is really small. You would never really interpret this as having a lot of a change in this pathway. But in this case, the directed score is also 0 because the upregulation balances the downregulation. The scenario I just showed you before is this one here, where you have genes that have, in terms of their magnitude of change, up versus down regulation, it's significant. Some are pretty strongly upregulated, some are pretty strongly downregulated. Our global score is decent, indicating a certain, there's some change happening in this pathway, but again, the directed score is 0. So just keep this in mind when you see values that are close to 0. It can indicate very, very little expression, or very little change, I should say, very little change in the pathway, or it could indicate there's a lot of upregulation, but that's also offset by a lot of downregulation. So that's a lot of math that I just went through. Hopefully that's starting to resonate with all of you. I think when we look at some real examples in the platform, it'll make a little more sense. So let me do that now. I'm going to jump to Rosalind, and let's walk through a data set and look at these GSA scores, but also what the intelligence capabilities in the platform bring. So bear with me as I transition over. Okay, so I have a public data set, and what we have is a ovarian cancer cell line. It's a human ovarian cancer cell line, and it's stimulated with, well, it's basically treatment versus control. It's anthraregulin versus BSA control. So let me show you what this looks like here. So this is the experiment. This is the public data set. So we have this cell line. Actually, let me go to our sample summary here to show you the experimental design. So in this case, we're going to focus on only these samples at the top. We're going to focus on these 6 here at the top. So all of the treatment is done at 2 hours time points, and we have 2 treatments. We have this, it's a recombinant anthraregulin treatment here versus our BSA control. And this happens to be in the cell line OVCAR8. This is ovarian cancer cell line. So we're looking at these 6 samples, this treatment, this recombinant anthraregulin versus our BSA control. So it's a very simple treatment versus control paradigm. And let's look at the results for this differential expression. So this analysis was done with NanoString's IO360 panel. So this is a panel focused on immuno-oncology applications here. And let's look at our results for differential expression of treatment versus control in our cancer cell line. So if you're new to the Rosalind platform, let me briefly introduce you to our interactive analysis page here. So what we have is a consolidation of our significant results here when we perform differential expression analysis. So on the left-hand side, we have a list of significant genes. What's really nice in the platform is that we have the ability to very quickly change our statistical cutoffs. So if you want to change your p-value threshold from 0.05 to 0.01, or you want to use an adjusted p-value threshold instead of an unadjusted p-value threshold, you can change the full change thresholds. It's really easy to do in the platform. So I have here created 3 different kind of scenarios, what we call a filter, which are basically just cutoffs for significance. So if I want to change those, I can just simply click on this and the whole screen updates at the same time. So in Rosalind, it's very easy to change your statistical cutoffs and refresh your view to get a most up-to-date view. And so in this view here, we're looking at a set of 31 significant genes on the left. We're visualizing them as a volcano plot here. We have gene-specific visualizations down here. This is our heat map, our standard clustering, showing us the relationships between our different samples. And on the right-hand side is where we start to see our pathway analysis and our pathway results. So what you'll see by default when you come to an N-counter experiment are all of our NanoString GSA scores. These are those gene set analysis scores. This happens to be the global score. So we can look at the directed scores as well in a different place, and I'll show you that. But by default, the first time you come to this page, you're going to see all of these GSA scores for your NanoString defined pathways. So these are annotated specifically, in this case, for the IO360 panel. So when you come here, you will see a list of pathways and their significance scores. So we know that there's no p-values associated with these pathways. We know that these are simply just scores trying to summarize how much change there is in each pathway, or how much fold change there is in each pathway, kind of conceptually, so to speak. So the way that you interpret these is simply, it's just a rank order of pathways. So myeloid compartment at the top with a score of 5.68, there's more evidence that this has more fold change in it, more change in it relative to cytotoxicity, which is on the bottom. So when you come to these NanoString GSA scores, it's simply a rank ordering. These are all relative to each other. How much change is there in a particular pathway? So what Rosalind brings is all of our knowledge and intelligence capabilities. So we have a big knowledge base that sits in the back end of Rosalind, which I'll show you in a few minutes here. And then we have a lot of intelligence capabilities. So our approach to get you the maximum amount of information out of your targeted NanoString panel is to bring in thousands of additional terms from pathways and signatures related to drugs and diseases and virology and ontologies. So we bring in thousands of additional terms and pathways and signatures from 50 additional knowledge bases to give you this extensive, expansive pathway analysis in the Rosalind platform, so you can extract more information out of it than these sets of pathways here, and also get novel insights that aren't going to be found in these pathways here as well. So let me show you what that looks like. So we can access all that additional information by clicking on this drop-down menu here. And these are the high-level categories of the additional knowledge bases, which are found in Rosalind. So let me show you some of these and some of the information we can get out of these. So let's go to our, let's say, pathway section. And here we can access databases such as Wiki Pathways, Bioplanet, Panther, Reactome. You may have heard of these before. And before I go into this, let's take a step back and think about the experiment we're looking at. So this is again, it's an ovarian cancer cell line treated with amphoregulin. So if you're not familiar with amphoregulin, it's actually a ligand for EGFR and the EGFR signaling pathway, that receptor. So this has a really important role in growth, cellular proliferation, inflammation, immune response, and it's also a really well-known player in cancer and cancer progression. So since we know the design of this experiment and the mechanism of action, it's a really nice positive control for the power of the Rosalind pathway analysis here. So what we find when we switch to our pathways databases is one of the first things that comes up is here, EGFR1 pathway. This is directly the mechanism of action of the drugs that we're testing. This is pulled out straight away from the Rosalind intelligence, the Rosalind extended pathway analysis. If you remember, we did not get that as part of the standard NanoString annotations because it just wasn't defined ahead of time. But using this kind of unbiased approach across 50 knowledge bases and thousands of terms, we can easily identify and pull in directly the mechanism of action of this particular drug. So that's a really nice kind of positive validation that what we're doing has a lot of value here. Some other things I want to show you is maybe let's look at one of kind of my favorite things is this transcription and regulation. So I study a lot of transcriptional regulation when I was in grad school, so it's always close to my heart. But what Rosalind can do is we can look at, for your set of significant genes, we can look at the promoter sequences, and we can identify, is there an enrichment in any transcription factor binding sites or any transcription factor motifs in the promoter regions of your significant genes. And so to do that, we can go to databases such as TRUST or JASPAR. And potentially here, we're not quite at the significance level, but potentially the XBP1 transcription factor is regulating some of these significant genes. So this is a nice piece of information you can't get from the standard kind of GSA score approach, where you can look at the transcriptional regulators of your significant genes. So if you want to know what those are, the great thing about all of this is that it's completely interactive. So any one of these terms I can click on. So let me click on XBP1. And what Rosalind will show you is that these three genes are potentially regulated by XBP1 transcription factor. Now, I didn't know about this transcription factor ahead of time, but I read a little bit about it, and it is actually implicated in cancer as well. So this is quite an interesting connection that I didn't know about this information. I didn't know about this transcription factor, but Rosalind brought it to the forefront, brought it to my eyes right away. And now I can start learning more about XBP1, its role in cancer, cancer progression, and some of its actual regulatory targets. So that's a really interesting piece of information that came out of here as well. So there's a few other things I might want to show you here before I move on. There's also this ontology collection, which is quite popular and useful here. If you're familiar with GO or gene ontology, this has been around for, I think, a couple of decades now, but that's also available within the Rosalind platform. You'll recognize that as biological process, cellular component, molecular function. Now, in this case, none of these terms were significantly enriched, but in other cases they are. It just depends on the context of your experiment. So Rosalind does contain gene ontology, which is one of those kind of best practices, gold standard pathway approaches for your data. So that's found here. Don't have time to go through all of the data, of course, but I'll show you a couple other highlights. We have this drugs category, which for me is really interesting because I have a background in drug development. So if we were studying potentially a new compound or a new molecule, and we didn't fully understand its mechanism of action, we could come to these drug databases and we could look at this and say, well, these known compounds have a similar gene signature, so they are significantly enriched in this case. So these compounds have a significant enrichment relative to my novel drug that I'm testing here. So can I infer that my novel drug, my novel compound, has similar mechanism of actions to these compounds here? Maybe they do, maybe they don't. These are ways to try to guide you to uncover what is that mechanism of action of your drug. So this is a really great category, a really great section for anyone working in drug development. That's one of my personal favorite sections here. So you can see as I'm going up and down here, there's a lot of information to look at, a lot of terms, a lot of databases, and now we can bring Rosalind intelligence in. So our approach here is to apply 50 additional knowledge bases with thousands of terms to give you this most comprehensive pathway analysis you can, but we want you to extract as much information out of it as you can. You certainly can go through all of these pathways one by one, and I would encourage you to do so you don't miss anything. But we want to make sure you don't miss any insights. We want to bring you all of the relevant information as quickly as possible to you. So that's why we created this Rosalind Intelligence. So if I click on Rosalind Intelligence, we have this nice summation of all of these 1,000 terms and all of these 50 additional knowledge bases. So what we're doing here is actually quite sophisticated, but we look across all of these thousands of terms for all the additional pathways, and then we cluster them together by similarity. So you can imagine there's a lot of similarity between the databases and the terms, and there's maybe overlap between the different genes. So part of Rosalind Intelligence is just clustering those similar terms and pathways together. And then what we do is we find out which of those clusters are the most significant, the most enriched, and then we pull out representative terms from all of those important clusters. So what we're seeing here in this enrichment summary is the result of that analysis. We've clustered all thousands of terms, we've removed redundancies, we've figured out what are the most representative terms and pathways across all those clusters, and those are brought to the forefront here in the summary. And this would be, in my recommendation, the place to start, because these are meant to represent everything else, all 1,000 terms plus in all of our databases. So when I come here, like if I was going to do this analysis on my own, I would start with this enrichment summary. I would look through these. I see this is interesting, rosiglitazone. From my background studying metabolic syndrome, I know rosiglitazone is the target of PPAR gamma, or it targets PPAR gamma. But this triggered me to actually read a little bit more about this. So I figured out that rosiglitazone actually targets the EGFR receptor as well, which is the target of our drug here, amphoregulin. So this was interesting to me. One thing that I see here is this A2F transcription factor network. This is really interesting. So let me click on this. And what we have is that all of these genes are potentially part of this A2F transcription factor network. So this is another kind of transcriptional regulatory insight that I would have missed if I wasn't doing this type of analysis in Rosalind. And then I also see your FRA pathway. This is really interesting. I didn't know about this. I had to do a little reading, but the FRA pathway is also involved in cancer as well. So I want to learn a bit more about the FRA pathway, the genes that are involved in this pathway, and that'll lead me to do insights and next steps for my analysis. So that is the our approach to pathway analysis in the platform, we want to take your targeted data, we want to apply 50 additional knowledge bases, thousands of terms, apply intelligence to it so you can extract as much information out of that as possible and get novel insights that you might miss if you were doing just the traditional GSA only approach. So that's the Rosalind intelligence approach to our pathway analysis. Now there's a lot more things I can show you. I'm going to take probably about maybe 5 more minutes and show you some additional components, and then I'll pause for some questions. Let me go back to, let's say, pathways. And you may have noticed that next to the name of each of these databases, such as Wiki Pathways, there's this magnifying glass. And what the magnifying glass represents is you can do a deeper dive on any one of these particular databases. So let me click on this magnifying glass here, and let's look at all of these Wiki Pathways results. So I'm going to click here and open up this page. And now in the table, we have the full results for pathway enrichment in Rosalind for Wiki Pathways. So we can get information about the adjusted p-value, number of genes in the pathway, number of genes significant, number upregulated, number downregulated. I can scroll through this table as well, like this. What else can I do? I can click on this download icon right here. I can download the results as a CSV. And what's really cool is we have pathway visualizations and pathway diagrams. So you've probably seen these before. So let's say I'm interested in cytokines and inflammatory response because I understand my drug here, amphoregulin, is important in potentially modulating the immune response or the inflammatory response. So let me click on this golden magnifying glass, and I'm going to open up a diagram of this pathway, and that's shown here. So we have a nice description of the biological function of this pathway here, and this is a nice visual representation as well. So these are so Rosalind does have the standard pathway diagrams that you've probably seen in other places. And what's great here is that we also have some interactivity. So you can hover over the genes on the diagram, and then on the left-hand side, you'll actually see what is the data. What's the full change? What is the p-value? You can modify how you are representing the data as well. So it's all nice and integrated. And then certainly you can look at a list of genes as well. Last thing I'll show you before I pause for some questions is some of the additional intelligence capabilities. So I'm going to go back to our interactive analysis page here. Let's say I am looking at my Rosalind intelligence summary, and I was interested in this FRI pathway. So let me look at all of my significant genes in the FRI pathway. And I recognize IL-6. I think I understand its function, but FOSL1, I don't know anything about this gene. And so this might be kind of a sticking point. You might have to go to like a Google search, look at 10 different databases to understand what is the role of this gene. But in Rosalind, we apply some intelligence and some generative AI to bring all that information to you directly in the platform. So FOSL1 is my significant gene. I don't really know much about its function, so I can click on the gene and I can click on my literature tab at the bottom. And so what we're doing here, and this is happening in real time, is Rosalind is scouring all of the known publications for FOSL1. You can think of this as like a PubMed search. And then we're applying some algorithms to it. So we look at all of the knowledge, the public knowledge, publications of FOSL1, and then we rank order the publications according to relevance, according to does it describe its biological function, how recent is the publication as well. And so we rank order these publications, we pull out the handful that are most representative, that are most relevant, that summarize the function of this gene. And then on the left-hand side, we apply generative AI. So on the left-hand side, we have these references at the bottom. So these were the highest ranked publications according to the function of this gene. And we applied a generative AI to make this summary about the function of the gene. So we can see that this is related to breast cancer, no surprise here, aggressiveness, metastasis, prognosis as well. We get a little information here about the regulation and function. So what's really important here and what's really neat is that this is always going to be up to date. This is completely novel. So if there's a new really important publication on FOSL1 tomorrow, and I ran this publication summary tomorrow to incorporate that, this publication summary would recalibrate. It would then it would incorporate this new information. So this is always going to be up to date for you. So this is a generative AI. You can think of it kind of like a chat GPT, but we put a lot of work in the background to make sure it's accurate. So uh, I think maybe 6 months or so ago, it might have been longer now, but there was a story in the news where a lawyer was writing a brief to submit to a judge for trial. And he actually used ChatGPT to write this brief. And ChatGPT actually used something called hallucinations. The ChatGPT actually made up cases that didn't exist. And the lawyer didn't proofread this, and he submitted this trial brief to the judge containing completely false and made up cases. So obviously that had huge repercussions for him professionally, but that's a cautionary tale, and we took that to heart. So what we did when we created this generative AI summary, we did a lot of validation and hard work on the back end to make sure there were no hallucinations, that it's completely accurate and up to date, summarizing the function of the gene. I'll show you one more thing, and then I'll pause for some questions. I know we're low on time. I do want to explain the knowledge graph. So what's sitting behind Rosalind is our knowledge graph. And what we've done, what our data science and AI teams have done, is we've gone to all of these public repositories, and we've integrated this information. So this is information about gene functions, different ontologies, representations, drug characteristics, cell types. It's this big wealth of information, billions of data points. And our AI team, our data science team, we harmonized all that information so it talks to each other. And so all of this can be directly accessed in the Rosalind platform through this knowledge tab here at the bottom. So this is the foundation of everything we do. You may not see it at all times, but it's always there. It's always informing your analysis and guiding you to get that interpretation, in this case, for pathway analysis. So there's 3 minutes left. I think I'll pause for some questions, but thank you all for listening.
Awesome. Thank you so much, Ryan. Let's get to the questions now. So the first question that we have is, where is the test statistic in our data? So I think maybe this is referring to the some of the stats that you were showing.
Yeah, okay, I understand. Yeah, so the test statistic, it's not directly shown as an output in typical analysis packages. It's not found in NSolver. It's not really found in Rosalind as well. So the test statistic is not visible. What is visible, of course, for your genes is the p-values. But the test statistic is just something that's calculated transiently on the way to generate the p-values. But they are used in calculation of the Rosalind NanoString scores here as well. So hopefully that gives visibility.
Awesome. The next question was, can we get PCA plots in the interactive analysis page?
So we have, we have our QC section, which has our MDS plots, which is multi-dimensional scaling. It's a data dimensionality reduction technique, very similar to PCA. So this is available not on the interactive analysis page, but on the QC section. So that's an interesting question and suggestion to integrate this into our interactive analysis. I'll have to think about that some more. So the question, I guess the short answer is it's not currently available on interactive analysis. It's in the QC, but that's a really intriguing idea to incorporate this on interactive analysis. So I appreciate that question, and we'll certainly think about this offline.
Awesome. The next question was, thank you, Ryan, amazing presentation. It's very informative. Can Rosalind analyze RNA-seq data from the whole transcriptome atlas obtained with the GeoMx?
And Kuki, if I could add, Ryan, the next question built on that from another person, can Rosalind work with DCC files? So these are obviously both related. Rock and roll, Ryan.
Yes, so yeah, this is a nice segue. So the answer is yes. So in the platform currently we have GeoMx analysis. So right now you are able to import normalized counts. So this would be normalized data that has been analyzed in the GeoMx instrument and the DSPA DA software. So you simply export normalized counts from that. You can analyze it in Rosalind, and you can get this full enhanced pathway experience as well. The question about DCCs is that is currently in development. So we have a wonderful new immersive experience for GeoMx, which is really gonna revolutionize how we do QC and how we analyze the data. So DCCs is coming soon. normalized counts is already available for analysis in the platform for geomics.
And Ryan, I'm going to add on that and just tell everyone from the CEO's voice, we're pushing as hard as we can to get that done this month. So everyone that either has an academic subscription or a NanoString professional subscription or an enterprise subscription, you will automatically get not only the advanced features for Encounter, but you'll get the geomics experience with the advanced geomics data analysis. It's awesome. Ryan and his team, having worked with thousands of geomics data sets, has made sure that we've built in everything you always wanted to do with geomics, but could never do on the instrument, or when you tried to do it, it was slowing the instrument down. So there's going to be a great experience coming to geomics, and hopefully, fingers crossed, working really hard about a month's time. So if it's not in a month, it'll be very shortly thereafter, but we're pushing hard to get that done. Kuki, I think you also had another question that asked about can Rosalind display bubble plots as a summary for both score and p-value, similar to GSEA for the biological function?
Right.
So, yeah. Okay. Yeah. So currently, it's not a feature that's available for visualization. We've received this request before, though, and it is certainly something that we are, developing. Don't have a timeline estimate for this, but I appreciate that question because we have received it before. We recognize its importance, so not yet.
And what I'll say on this one, and by the way, thank you for the question. I know the person well who asked it, and it's great to hear from you again, and I'm glad you attended today. We are working really hard within the intelligence team to bring two very important updates that will enhance everything you saw today, and this is coming soon. The first part I want to mention is that we'll be regrouping the pathways around the biological entities. So no longer will you have an option effectively of being able to see things grouped by knowledge base or see things grouped by biology. So the biological representation is coming soon. The knowledge bases will be updated, and the speed with which enrichment is performed is going to be radically accelerated. So, and the Rosalind Intelligence feature will apply not just to the collective of all the knowledge bases, but it'll apply to each biological area. So you'll be able to see the non-redundant, non-overlapping important terms for each respective biological aspect. So that is close at hand from the intelligence team. With one other very important update, we already have 15,000 major public data sets ready for correlation. And we will be showing you in the same interface the ability to see how your data, based on the expression of your information, relates to or anti-correlates to available public data. So this is the next level of deep insight. And the reason why I really want to highlight this before I have to jump to my next call and leave Ryan and Kuki to wrap this up for you all is that your data is never static in Rosalind. The intelligence and the interpretation is always growing, it's always expanding, and there's always new insights available. When you finish an analysis, you should never think about mothballing it and not coming back. Of course, you need to use it in meta-analysis, which Kuki and Ryan are going to tell you is the last session of the Masterclass series. But things like these intelligence enhancements allow you to go back and find new insights in your existing data. So it's part of the ongoing value of the Rosalind platform. So I'm really excited to see these upcoming capabilities coming. So to this question about bubble plots and GSEA, it's a really great request. And as the intelligence team adds more and more capabilities and expands our bottom bar, as we call it, the intelligence bar, that could be something that we get added in that area. So please keep all the suggestions and feedback coming. Anyway, Ryan, I got to hand it back to you. I'm late for my call. So great session.
Awesome. Thank you, Tim. Looks like we are over time. Maybe we didn't get to one or two questions, so we'll make sure to get those back to you via email, just so that we keep everyone on time. Thank you so much for attending the session today. And as Tim said, we have 2 more sessions remaining in the master series. I can't believe we're already towards the end. So hopefully we'll see you in 2 weeks for the cell profiling series, which is on 13th of June. And then the last one on meta-analysis will be on 27th of June. So hopefully we'll see you guys in June. Thank you so much for attending today. Have a wonderful day. Thank you.